So I just want to thank you for taking this time. I know your time is very valuable. I just wanted to ground this in the original email I sent because it's genuinely the center of what I'm building. At its core, Enigma Genetics is a infrastructure effort, identity, custody, consent, and provenance in long-term stewardship of genomic and epigenomic data. I'm not coming to you with a biological breakthrough. I'm coming with a systems questions. So yeah, I just curious about if you had any time to think about this and what your initial thoughts were. And then, I really want this to be a conversation. I'll go into my background. I want to be very transparent. I want to show you my own limit. So I'm an engineer by training, not a genomist. I expect there are gaps in my thinking, and some of those gaps might not be fillable. That's why I'm really coming to you with some of these questions.
What you're talking about here resonates very much with me. We developed personal genome project to do some of the sorts of things you're integrating genotype with phenotype, maybe environmental influences, bridging the two. We did include brain imaging as one of the modalities, fMRI on many of the volunteers. Randy Bruckner at MGH, I think, did the study. I'm not sure it ever got published, but this is easily a decade ago. The whole personal genome project is now 20 years old this year. We are interested in another kind of brain imaging, which is destructive. I mean, MRI is perfectly benign, but we're working on connectomes, and we have a new method of doing connectomes that with synapse level resolution, we think we get at least the whole mouse brain probably in a year or so, and we'll scale up to human. But that would be autopsy,
Hmm.
at least the methods we're using so far.
My intuition, we treat this data like a snapshot rather than a moving film. To me, that if we could have one of these snapshots as almost a version control type
Yeah.
situation, right? And have that as, there's a lot of potential for AI and modeling. I would even go as far as to say long-term thinking is restoration paths. So what are your initial thoughts on that statement?
The statement is about restoration.
It seems to me that most of the studies have been done against the population rather than the person, right?
That was one thing that distinguished the Personal Genome Project. It is aimed at the individual. To simulate the future physician and patient experience of most databases right now, they're de-identified kind of ensembles of genes and phenotypes. This would be one where you have a data hairball of all the genotypes and phenotypes of a particular person. That's the unit upon which a physician works, maybe with a little bit of extra data about the family, but mostly about the individual. That's just not feasible with most of the resources that include genomics.
When you say not feasible, just the cost.
No, I mean, they've been structured with such an extreme emphasis on privacy that they'll basically give you allele frequency for phenotype X, and you have no idea of whether that's correlated to phenotype Y or Z or environment. You really want to have all of that, because if you want to build a system model of a person, you need to know a great deal about that person in a way that everything is mappable on everything else. And the problem is that the more unique features you integrate, the more that person becomes identifiable. I pointed out in 2005 that we already crossed that threshold as soon as we start doing genome sequencing. Genome sequencing alone, forget about all the phenotypes, it's identifiable.
That's exactly what I've been thinking. It's mostly on perception and the way people are thinking about this that really hinders a lot of possibilities. So with that being said, it could be extremely important. I'm still building a lot of this in my brain and bouncing it around. I'm envisioning the most perfect version of ourselves, right? Maybe even in utero, we're taking DNA samples and mapping as the influences of our environment in real time, creating that database for science later. Because we don't know how to do all the tests and we don't know everything we're going to need, but we know the possibilities are on the horizon. And I think as this becomes more important, more money will flow in this direction. What I've seen and experienced, I want to pick your brain on what you think the main pitfalls are here and where you see holes in this.
Well, I think I see more opportunities than pitfalls.
That's encouraging for sure.
I think some of them are immediately available. One of the problems with this field is we've talked so much about the promise and how you got a big data set, you could do awesome AI, even before AI association studies. We got so enamored of that far off, complicated thing that we somehow forgot that we could save trillions of dollars just by solving Mendelian diseases. And that doesn't require new data, barely requires new software, doesn't require infrastructure building. We already have enough worldwide to sequence everybody's genome. And if we don't, it would be a trivial scaling problem. We're way past the point of profitability. That is to say, we don't need to worry about who's going to pay for this. It pays for itself. And it doesn't require that big a startup investment in infrastructure. Most of the infrastructure is all the sequencing devices that already exist worldwide.
It's about partnering and putting it together.
Right, exactly. So the simple equation in engineering is that it costs about $1 million per person if you have a Mendelian disease. The fraction of those in the population, about 3%. It costs on the order of $300 to get your genome sequenced and interpreted, and that would be the 97%. Only 3% would benefit from it. They would be able to make some decision that would help them have healthy babies. If you start with 100 people, they'll all pay $300, right? So that's $30,000. And then 3% of them, say 3 out of the 100, will save society $3 million. You paid $30,000 in order to save $3 million. That's about a 100-fold return on investment that you can do with available technology, with available infrastructure. You don't have to build anything. You just have to collect the specimens, send them out. You take responsibility for the medical. It's like an insurance company or a state-run medical care provider. To me, it's a no-brainer, but nobody has implemented this yet. They've implemented all the parts, but they haven't put it together into something where you do better than break even.
That's incredible. I've thought a lot about taking the existing sequence data, whether it's even 23andMe, and starting to build models off of that, right? So
I would not recommend 23andMe. I've been involved in 23andMe since before they started in 2006, serious diseases. They were designed for finding, ancestry, for finding common allele associations, but most associations for really important diseases, rare alleles, and they're not on the chips. You need whole genome sequencing for that. Whole genome sequencing, I simultaneously helped with getting 23andMe started and Gnome. Gnome charged $350,000 per whole genome sequence. 23andMe charged $1,000. And so there was, they were obviously aiming for different markets. Now it's basically $300 for both, which is less than either of them originally, but if anything is converged on whole genome sequencing is cheap enough, you might as well do it.
Yeah, that's been my perspective on it too.
Given whole genome sequencing, there's a lot you can do, especially if you have phenotype and genotype together,
Overlaid.
like EGP.
One thing I've thought a lot about is how I could compress this data into a meaningful and useful way that would help the field. With that, doing reversion control in notes, here's the changes from A to B, and only noting that. We have one data set with just those revision notes.
Yeah, that's been implemented. So the highest compression I've seen is about 3 megabases per genome. So technically a diploid genome is 6 billion base pairs, going from 6 billion ACCGs and Ts down to 3 megabytes sounds like a pretty good compression ratio.
Absolutely.
You could fit entire towns into a typical laptop drive.
Is that in reference to human DNA? So potentially we could.
That's purely prefaced on having a reference genome for humans,
That's what I was.
and then
Okay.
just looking at the differences between your diploid genome and reference.
Okay. So that's what I was envisioning.
Now, you could do that for any species, but
Right.
each species would have to have its own reference.
Right.
And some species are much more diverse than humans. So, for example, chimpanzees are much more diverse than humans are, ironically. Even though there's 8 billion of us and 100,000 of them have more diversity.
If we were to look at humans and maybe lump them into groups, if you moved a person into that, there's a smaller fraction of changes. You could potentially say,
Oh, yeah, I know what you're going. I think there is a slight
but it's not.
advantage there.
You
Not,
lose
yeah.
data by the compression. Understood.
And then the other thing you have to think about is compression of the phenotypes. That's not a solved problem. It's not a challenging problem necessarily, but it's just not solved. Genome one is a very easily stated problem, and anybody familiar with compression would see the opportunities. For phenotypes, we still don't have any kind of standardization except a few that are in routine medical use, like imaging is well. There's fairly limited number of ways of representing imaging data, most of them proprietary. Those are big data sets. Most other phenotype data sets are pretty small, like control vocabulary on a medical record of whether you have autism spectrum disorder or something like that.
Right.
There's usually not a lot of detail about that until you get to imaging, or, you have a red blood cell count or lactate levels. All those things are very, very depressed already.
Okay. So I guess I want to pivot a little and ask, going back to, I mentioned versions and creating a version log. Say that you were to have a reference from 10 years ago. What would be the challenges of actually making that reversion? And is that something that's actually
version or reversion?
Reversion? So we have a known good sample, and maybe even two of them say you're in a good health, right? And then all of a sudden you go down. We take another snapshot and there are some clear deltas that are absolutely different. If
We're surrounded by AI, I think that's an unsolved problem. It may have computational components, but it's mostly an unsolved biological problem. A computer could probably calculate what you would like to reverse. Scientist, physician team has to figure out how to actually implement that.
Right. My question, say there were clear markers, how many changes over time are there? Is it millions? Is it hundreds? Based on what you've experienced and seen.
There are markers
The
that are
methylation.
not necessarily causative. There's bystander markers for making clocks of biological age versus chronological age. It's become a pastime for people to try to carve off their biological age.
Right.
And they could tell all their friends, hey, I just had my 50th birthday, but my biological age is 40. So that's there's most of those are non-causative as far as we can tell, things like DNA methylation. Then there are things which clearly are in the causal pathway, so for example, loss of muscle tone, various frailty indexes, pre-cancer and cancer, things that are starting to stack the deck against you, but aren't really killing you. Most of these fall under the umbrella of aging.
So that's where I'm going with this. It seems to me that it's almost impossible to map what the causation is based on the plethora of changes.
I don't use the word impossible, but I would say it's very difficult today, but in the future it might be easy.
Fundamentally, what I'm talking about is maybe not caring about what all the changes are and what they mean, because we know we had a good state. So can't we just go back to that without understanding everything?
Well, the simple answer is yes, at least for certain cells and tissues. For example,
that's the uptake limitation, right?
If you wanted to replace your blood with an earlier version of yourself, and in fact, people do this for short periods of time, but you could do it for long periods of time. For example, they'll have some hematopoietic cancer, and then destroy the whole blood production system, and then bring in either from a donor or from yourself, earlier version before you got the cancer. And then it will repopulate typically the bone marrow, and it'll start producing all of the white and red blood cell lineages. That is done. Now, it's not typically done as a rejuvenation thing. It's mostly an anti-cancer thing, but it could be rejuvenated. It's only worth blood. If you wanted to rejuvenate, let's say, your kidney or your liver, then you'd have to get a transplant.
or potential sample.
You could get a transplant from a young person or a young pig. We're working on various drugs and gene therapies that will make your liver younger. Your liver produces almost all the proteins in your blood. If you have a young liver, you have a good chance of all the proteins in your blood being young. Since those go everywhere in your body, there's a chance it has a general rejuvenating effect. We've done preclinical trials with animals and shown we can train the liver to produce rejuvenating factors that work on large parts of the body, a longevity effect. You actually have a survival curve that's shifted to the right.
Are there any?
There's no general solution to it that I know of.
So, say if you were going to try to gather data points from all the cell types in the body to have your version control, would you be able to do that? Would that mean one from the brain, one from the heart, one from the lungs, one from the liver?
You can certainly collect the data. You could plan a strategy, but certain tissues are easier than others. I mentioned I started with the two easiest ones, which is blood and liver. Brain is probably the hardest one because it depends on all the 8,000 synapses per neuron, everybody connected to everybody. If you take that apart and replace it with something else, it's no longer you. You won't sound like you. You won't think like you. You won't fool anybody.
So that could be the limitation.
And most organs fall somewhere in between. Some of them are inconvenient to do surgery on.
So, like with the liver as an example, are there any other qualifiers that you are looking into, or is it just a healthy, good liver? Does it have to be the same blood type? Or
transplants?
Yeah, right.
Most people that are looking for liver transplants right now have hepatitis or some other serious, alcoholic's liver, or they have a gunshot wound, and they're just trying to replace it. Usually the liver disease is so sudden in its onset that you don't have time to find a donor and then you die. But we're working on ways to bridge that so you can get an immediate liver replacement and then line up a more permanent one.
Where would you point me to go next with this? I'm definitely needing some more mental clout and some people with a great understanding to think about this and maybe talk about it and make it better so I could actually bring it to market or potentially try to do something like this. What would you say to that?
How fast do you want to get to market?
I mean, I'd love to do it today and that'd be my full-time gig, but yeah.
Yeah, so I think you're going to have to start with something quite a bit short of reversion.
Oh, I absolutely know that. And that's my plan was to somehow get a data structure in place and start changing the way people think about this.
Taking it from a data standpoint first, setting up not sequencing infrastructure, but data infrastructure. I think one of the things that would reassure people is privacy. But if you have too much privacy, then you can't ask questions about individuals. There is a growing literature on being able to ask questions without re-identifying. You could have an encrypted data set for an individual and ask questions about it without decrypting it. An example of that is homomorphic encryption queries. Those are typically limited, especially if you've got a large community asking those queries. If you ask enough questions, you're going to start getting personal information out. I mean, you'll have a bolus of information that collectively identifies the individual. So there has to be limitations. That's one solution to the privacy issue. The other end of the spectrum is the Personal Genome Project, where we just consented people for disclosing their identity, either intentionally or inadvertently in the process of sharing lots of data. I think we probably need both of those. Different people are going to be reassured by different things.
It's almost elective. That's what I envision is bolting this onto a sequencing lab and then just saying, hey, You want to opt in on this?
And so the third option they have, one is complete encryption, never decrypt it. Another is complete openness. And the third is not participating in the research at all. You get your genome, have your own encrypted version that you can share with your physician, but you're not sharing it for research. You're not sharing it for, it's not for sale, so forth. So there are at least three versions. But you're right. Since the genome pays for itself, you don't need to worry about income from research. You do have to worry if only 1 million people sign up and you sequence 8 billion, it seems like a fairly low yield, but at any point, something could be a sea change where the 8 billion look at the 1 million and see they're getting better health care, that they're heroes, that they're participating in science, and then they may convert over. It tends to be a ratchet in that once you start donating your medical data to science, you keep doing it. And especially if you release it on the internet, it's really very hard to go back.
If we can make this some kind of monetization to the consumer, right? So as their
Right.
information is being utilized in studies, they can elect in and maybe even go onto a marketplace and say, hey, I want to do this.
Well,
this.
one way to prime that pump is the business model I mentioned at the very beginning, where you're eliminating Mendelian disease. You could give them a cut of that right up front. My estimate is about 100-fold return on investment, meaning save $3 million by spending $30,000. You can give them a cut of that. If there is a drug that's invented based on their genome, it'll probably be based on thousands of genomes,
Right.
but they'll get their 1,000th of a cut.
And it becomes easier to engineer every drug by the masses that you get into this data set.
I
right?
mean,
There's
for
so many-
example, you can do pharmacogenetics to stratify who should get it and who shouldn't get it, who it's going to be toxic for, who it's going to be saving, and who it's going to do nothing for. Got price tags attached to it as well. We tried to do something like this marketplace in one. I've tried many different business models over the years. This has been my dream since the 1970s. And one of them was called Genos, where it was a marketplace where you could sell sequencing capacity, you could sell sequencing storage, you could sell your genome to, or genomes you've collected with phenotypes, without phenotypes. I think it was a good idea, but it didn't catch.
Well, it's just too early. Timing
Yeah,
is
absolutely.
everything. And one of the things you mentioned 23andMe and how that was kind of
Too early
yeah.
and not configured for medicine.
Wondered, it seems like as you got more data, it would probably point towards 23andMe getting worse, right? They
That's
made a
right.
lot of
As you get-
assumption as they added data to it, they actually were corrupting their own information or the results.
I'm not sure it's corrupting. They were just
Yeah.
limited. There just wasn't enough information. And AI can't fix that either.
You're right.
I
You're right.
mean,
You can't fill it in.
You can squeeze a little bit out, but the fact is there's serious missing data.
Yep.
There have been proposals that if you sequence a couple of family members from each family, do sparse sequencing of the rest, you could infer it. You can't infer it in the big population. You could infer it at the family level. But again, it's just not worth it. The cost is about $100 a genome. Whether it's 100 or 300, it still is too cheap to meter. The return on investment is so high, you might as well just do the whole genome for everybody.
What is the cost to run the epigenetic?
The problem is, unlike sequencing is very defined. You have 6 billion base pairs, and you pretty much sample any part of your body and get the same answer. There's a little bit of somatic mutation and mosaicism where your right hand might be different from your left hand, but it's mostly very well defined. Epigenetics is the opposite, almost. That is to say, there's more than one kind of epigenetics. Like there's only one kind of genome, RNA epigenomics, and there's protein and carbohydrate, and there's morphology. It's almost all of phenotypes falls in epigenetics. But even if you're restricted to DNA methylation and RNA, then you have the problem of which cell type. Most people implicitly assume you're talking about blood cells, white blood cells, because they have genomes.
Yep.
And they're very easy to get. Might be able to do with saliva cells. Saliva, one thing that not many people know is saliva has almost exactly the same amount of DNA. A mill of saliva has the same DNA as a mill of blood. Most people think that blood is just chock full of DNA-containing cells. The saliva is mostly mucus, salts, and water. There's lots of cells in there. I haven't seen that being used commonly for epigenetics, but you could do it. That leaves out all the other cells that determine whether you're sick or not. For the most part, your saliva and your blood don't determine that you're sick unless you've got some immune deficiency or
Blood
viral
disease.
infection or something like that. But the brain and the liver and the heart have their own epigenetic signatures, and they're hard to get. Nobody wants a biopsy, just a tiny biopsy from each of your organs.
But it is useful. It could be extremely useful, right?
But just imagine what kind of surgery that would be to get a little bitty bit of 300 different organs.
Yeah. No, absolutely.
While you're alive. One possibility is you can get some of that information from circulating the blood, but it's hard to tell which tissue it actually came from. It's much better to go in there with like laparoscopy. You've got a very tiny incision. You snake this thing in and you take a little bit, come back. That could possibly be an outpatient procedure, but
It wouldn't be
it's
fun.
hard to get permission to do that because start with a patient that's fairly healthy and you're looking for trouble, and you'll probably find it.
That's the truth. It's so personalized and specific that any inference you put in, you're vulnerable. You're playing the statistics. I dated a girl who was set on having her breast removed because her mother had breast cancer. And I had to pray. It's like, no, do not do that until you have the first sign of it. Does not make sense to have your breast removed today, right?
Yeah.
With 20% their likelihood of getting cancer. Right?
People have different risk tolerance. Some people feel the risk of dying of tobacco-induced disease is low enough that they chain smoke their whole life.
Right.
Other people are unwilling to leave their home. 1 million people die per year in automobile accidents.
Right.
So it's hard to say. Some people don't think their genome is a risk. My genome is public information. Other people, clean up after themselves, like presidents of the United States, they go around cleaning up so there's no DNA left behind. I have only 3 minutes left before my next appointment.
Would you be open to potentially talking again after I take this information, circle back and clean it up a little bit?
Yeah.
Okay.
Sure.
Is there anybody you could think of that might be interested in helping or putting their brain on this and seeing if it resonates with them?
There's a company called Curie, which I co-founded many years ago. It's part of the Personal Genome Project. They are putting together a little box which has GPUs, artificial intelligence, and all of the genomes that we've collected. It can be a test bed for a variety of things. I'm sure you might find some kindred spirit in that operation. I don't know whether they have a website or not. Curii.
Okay. Curii. I'm working on some NIH grant funding. Once I get this cleaned up a little bit, I might have you take a look at it before I submit it, if you'd be willing to potentially do that.
Yeah.
Okay. I know you're a busy man. I'll let you be, but I really appreciate this.
Okay.
This has been an absolute pleasure, George.
You bet. Good luck on this.
All right. Thank you.
Take care.